Hello folks, my name is Logan Thrasher Collins, and I'm going to tell you about some very cool nanotechnology research from the Elbaz laboratory at Tel Aviv University. Specifically, we're going to be learning about a strategy for encapsulating DNA nanodevices inside of virus-like particles. The research that I'm going to discuss was published in the scientific journal ACS Synthetic Biology during 2021. Here is the title of the paper for those of you who are interested in looking it up and learning more. To start off, I'll give some background. It is possible to create DNA nanodevices by producing a sequence of DNA which folds into desired secondary or tertiary structures, similar to proteins. DNA nanotechnology in general has enormous potential for a wide variety of applications. However, DNA nanodevices are susceptible to degradation by enzymes in biological environments. This makes it difficult to manufacture them at scale inside of cells. This paper took a clever approach to protecting DNA nanodevices from degradation during cellular production. Since the bacteriophage MS2 packages RNA, which contains a hairpin loop called tRNA, the authors used an analogous tRDNA hairpin to facilitate encapsulation of DNA nanodevices into empty MS2 capsids, which were then referred to as MS2-like particles. To demonstrate this, the authors started with an in vitro method. They expressed MS2 capsid protein dimers in E. coli and then purified them, synthesizing a chosen DNA nanodevice equipped with a tRDNA hairpin separately. They next packaged the nanodevice in vitro by mixing it with the capsid protein dimers. Next, the authors tested the ability of the MS2 capsids to protect DNA devices from the environment. They chose a DNA aptamer which binds a dye called malachite green and enhances its fluorescence. When the DNA-degrading enzyme DNase-1 was added to naked aptamers, the dye's fluorescence went down dramatically. By contrast, when the DNA aptamers were protected inside of MS2 capsids, the dye's fluorescence was better preserved. The authors also showed that pH-sensitive DNA nanotweezers could function inside of MS2 particles. These nanotweezers stay open at a pH of 7, allowing fluorescence from an exposed fluorophore, and they close at a pH of 5, turning off the fluorescence by moving a quencher into the proximity of the fluorophore. Transmission electron microscopy was employed to directly visualize the MS2 capsids and their DNA nanodevice cargoes, leading to these very nice images. To develop a way of mass-producing the MS2-like particles, genes for the MS2 capsid proteins were expressed in E. coli alongside genes encoding DNA nanodevices with tRDNA hairpins. The resulting MS2-like particles were subsequently purified from the bacteria. Here is some more detail on the in vivo production process. The nanodevice encoding gene first transcribes an RNA with a reverse transcriptase binding motif. The bacteria furthermore express reverse transcriptases which convert this RNA into DNA. Part of the resulting DNA sequence was designed to fold into a DNAzyme, which cleaves itself and the reverse transcriptase binding motif away from the rest of the nanodevice sequence. The E. coli also express both the MS2 capsid protein and a protein called matrase, which aids the assembly of the capsid particles. Through the presence of their tRDNA hairpins, the nanodevices are packaged into the MS2-like particles. Here is a transmission electron microscopy image of bacterially manufactured MS2 virus-like particles carrying DNA nanodevices. To verify the functionality of the in vivo produced virus-like particles, the authors used MS2 capsids carrying DNA aptamers and the malachite green dye. They observed that the bacteria themselves fluoresced under these conditions, indicating that the capsids were indeed protecting the aptamers and allowing them to induce the dye's fluorescent state. Finally, the authors wanted to see if their invention might be useful in a biomedical context, so they tested the protective capacity of the MS2-like particles in human blood serum. When DNA aptamers with malachite green were put into the serum without protection, they quickly degraded and lost fluorescence. By contrast, the fluorescence was preserved in the case of the MS2 capsids carrying aptamers and malachite green, demonstrating that this system could be useful for transporting DNA nanodevices inside the human body. Thanks for listening. I hope you enjoyed my discussion of this exciting research.